Welcome to this explainer. Today, we are diving straight into the absolute engine room of our modern world, biotechnology. If you've ever stopped and wondered exactly how the microscopic realm fundamentally shapes our massive everyday human lives, well, you are definitely in the right place. Let's just jump right in. Okay, let's dive into this. Think about it for a second. What could a simple loaf of bread, that box of laundry detergent sitting in your cupboard, and life-saving human insulin possibly have in common? Right? On the surface, they seem completely unrelated, but the actual answer is that every single one of them relies on microscopic workers doing just extraordinary things. Seriously, these little organisms are literally the unseen workforce of our entire global economy. Now, before we go any further, let's quickly lock in our definitions so we're all on the same page. Biotechnology. It's really just putting biology to work. It's applying biological organisms, or systems, to manufacturing and service industries. Simple enough, right? Then we have genetic modification. This is a much more specific tool in that biotech kit. It's the actual physical transfer of genes from one organism to another. And often, this happens across entirely unrelated species. So here's our game plan for today. 1. The basics of biotechnology. 2. Everyday enzymes. 3. Scaling up with fermenters. 4. Editing the code. And finally, 5. We'll weigh the ecological calculus. All right, Section 1, the basics of biotechnology. To build our foundation here, we really have to ask ourselves, why are bacteria the absolute rock stars of the biotech world? Well, the answer comes down to elegance and efficiency. You see, bacteria have these small circular pieces of DNA called plasmids. Think of plasmids almost like open source software that scientists can just go in and edit. Plus, they reproduce incredibly fast, they take up virtually no space, and, this is the crazy part, their genetic code is universally shared with all other life on Earth. Oh, and growing them in massive quantities totally avoids the ethical hurdles you'd run into using higher order animals. Moving right along to Section 2, everyday enzymes and applications. So you might be wondering, how is this stuff already at work right in your own home? Well, consider yeast. It's a single-celled fungus, and it undergoes this process called anaerobic respiration. Basically, when it consumes glucose without oxygen, it spits out ethanol and carbon dioxide. And wow, is this an incredibly versatile equation? We use that ethanol to make modern biofuels to run our cars today, right? But bakers have actually been using this exact same chemical reaction for literally millennia. The carbon dioxide gets trapped in the dough, and boom, that is exactly what causes your bread to rise. Now think about pretty much everyone's least favorite chore, doing the laundry. The stains we get on our clothes are usually organic molecules, fats or proteins. Now, standard soap needs blistering hot water and some serious physical scrubbing to actually get them out. But biological washing powders, they kinda cheat the system. They use enzymes that are almost identical to the ones inside your own stomach right now to actively digest and break down those fats and proteins. They literally do the heavy lifting for you. This lets you wash your clothes effectively at much lower temperatures, which, as you can imagine, saves a massive amount of energy. And this brilliantly illustrates another super common everyday application, lactose-free milk. Since so many people lose the ability to naturally produce the lactase enzyme as they get older, manufacturers they add it right into the carton. The enzyme acts like a pair of microscopic chemical scissors. It cleanly snips the complex lactose sugar into two easily digestible simple sugars, glucose and galactose. Boom, problem solved, entirely by biotechnology. Okay, let's jump into Section 3, scaling up with industrial fermenters. Now, it's one thing to have a neat, useful little reaction happen in a loaf of bread on your kitchen counter. But how do we supersize that for the entire globe? Well, this all really kicked off when Alexander Fleming discovered penicillin back in 1928. Suddenly, we desperately needed a way to mass produce a microbe. Enter the industrial fermenter. You can think of these as basically 5-star microbial spas. They are gigantic, highly engineered stainless steel vats that let us culture microorganisms in truly colossal amounts. Seriously, every single water jacket, stirring paddle, and input pipe is meticulously designed to keep these microbes perfectly happy so they just never, ever stop producing. But to keep these living biological factories churning away without, dying off, the internal conditions inside the vat have to be rigorously, and I mean rigorously, controlled. For instance, with penicillin, the temperature is locked in at exactly 26 degrees Celsius. And actually, the vat generates so much of its own heat from all that biological activity that it requires constant cooling. On top of that, the pH is kept slightly acidic, completely sterilized air is pumped in so we don't get any foreign contamination. And the mold itself is fed this constant, never-ending buffet of molasses or corn steep liquor. So yeah, it's a massive, massive scale balancing act. Which brings us to Section 4, editing the code of life. So far, we've seen how we grow natural microbes, but now, now we're entering a whole new territory, actively manipulating their literal genetic blueprints. Think of this process almost like a highly technical recipe for, well, hacking life. First, we isolate a very specific gene. Let's say it's a human one. Then we use those biological scissors we talked about, specifically called restriction enzymes, to cut the DNA. And we purposely leave these jagged, unpaired edges that scientists call sticky ends. Next, we take a bacterial plasmid, cut it with that exact same enzyme so its sticky ends perfectly match up. And finally, we glue the whole thing together using another enzyme called DNA ligase. Just like that, we've literally pasted human code right into bacterial software. Now, what's really, really interesting about this whole cut and paste cycle is just the sheer elegance of it all. Once that DNA ligase glues the human and bacterial DNA together, we're left with what's called a complete recombinant plasmid. We take that, insert it back into a bacterium, and drop it right into one of those massive industrial fermenters we just looked at. And as that bacterium multiplies by the billions, every single new clone acts as its own microscopic factory. And what is it making? It's producing the exact protein coded by that inserted human gene. Mind-blowing, right?
And the real-world results of this technology, they are absolutely staggering. I mean, since 1982, we have used genetically modified bacteria to produce pure human insulin. Before that, insulin actually had to be harvested from pigs or cattle, which sometimes triggered really bad allergic reactions in people. We've also created things like golden rice, where a gene is inserted to drastically increase vitamin A levels, directly fighting child blindness in developing parts of the world. And in agriculture, we've developed crops armed with Bacillus thuringiensis toxins. I know, big word, but it basically just makes the plants naturally resistant to insect larvae. We even have modified wheat that is designed to be completely 100% free of the gluten that causes severe food allergies for so many folks.
All right, let's look at our final part, Section 5, the ecological calculus. Because, as you can probably guess, with this kind of great power comes massive, massive complexity. It really forces us to weigh the ecological calculus of these technologies. And I'll tell you, the ledger is pretty complicated. On the benefit side of things, GM crops that resist pests drastically reduce our reliance on spraying external chemical pesticides. That's a big win. Plus, drought-resistant crops hugely increase our land efficiency, letting us produce way more food without having to clear out precious wild habitats. But on the flip side, the risks are equally profound. There is a very real danger of cross-pollination, where herbicide resistance gets transferred to wild plants, accidentally creating these invincible superweeds. Furthermore, if we start relying on just a few dominant GM varieties, it seriously threatens global biodiversity. We run the risk of natural wild plant species just being outcompeted entirely.
So, what does this all really mean for us? Well, it brings us to the ultimate million-dollar question. How do we balance the immediate, truly desperate need to feed a rapidly growing human population in the middle of a changing climate against the unknown long-term impacts that these newly combined genes might actually have on our global biodiversity? It's a profound ecological calculus, and it's one that scientists, farmers, and our entire society are actively trying to weigh right now. I really hope this explainer has given you the solid foundational knowledge you need to jump in and engage in that debate. As always, keep questioning, keep learning, and hey, thank you so much for joining me today. Catch you next time.